Good evening, class. Welcome back to Microbiology. This is Silvana O'Coney, and in today's lecture we're going to take a look at prokaryotic reproduction and growth. First, we'll focus on binary fission, which is the type of cell division most frequently observed among the prokaryotes. Cell reproduction leads to an increase in population size, so we're going to consider population growth. Then we'll take a look at the influence of environmental factors on microbial growth. So let's start by considering the term growth. Growth may be defined as an increase in cellular constituents. It leads to an increase in cell number when microorganisms reproduce by processes like budding or binary fission, but growth can also result when cells simply become longer or larger. For example, if the microorganism is cenocytic. A cenocytic microorganism undergoes nuclear divisions that are not accompanied by cell divisions. So, for example, some fungi consist of hyphae that are cenocytic, that is, they're multinucleate. But it's not usually convenient to investigate the growth and reproduction of individual microorganisms because they're so small. So microbiologists normally follow the changes in total population number. The cell cycle is the complete sequence of events from formation of a new cell through to the next cell division event. Most prokaryotes reproduce by simple binary fission, although some reproduce by budding, fragmentation, or other means. Binary fission is a simple type of cell division process consisting of two pathways: DNA replication and partition, and cytokinesis. The process of binary fission is illustrated here. In A, we have a young cell at an early phase of the cell cycle. In B, the cell prepares for division by enlarging its cell wall, cell membrane, and overall volume. In C, the septum or cross wall begins to grow inward as the chromosomes move towards opposite ends of the cell. Other cytoplasmic components are distributed to the two new developing cells. In D, the septum or cross wall is synthesized completely through the cell center, and the cell membrane patches itself so that there are two separate cell chambers. At this point, the daughter cells are divided. Some species separate completely, as shown here, while others remain attached, forming chains or other arrangements. So it's important to keep in mind that this is a very simple form of cell division, very, very different to the processes of mitosis and meiosis that you're already familiar with. The cell cycles of E. coli and Bacillus subtilis have been extensively studied, and our understanding of bacterial cell cycles is based largely on these studies. This timeline shows that E. coli requires about 40 minutes to replicate its DNA and partition those DNA copies, and about 20 minutes after termination of replication to undergo septation and divide into two new daughter cells. So approximately 60 minutes in total. So we've seen that the cell cycle in E. coli takes approximately 60 minutes. But in rapidly growing E. coli cells, the cycle can be completed in only 20 minutes. How is this possible when DNA replication always takes about 40 minutes? E. coli does this by beginning a second round of DNA replication, and sometimes even a third or fourth round before the first round of replication is even completed. Therefore, the progeny cells receive 2 or more replication forks, and replication is continuous because the cells are always copying their DNA. So, binary fission and other cell division processes bring about an increase in the number of cells in a population. And this population growth is studied by analysing what's called the growth curve of a microbial culture. And this growth curve is observed when microorganisms are cultivated in batch culture. That is, they're grown in a culture within a closed vessel with a single batch of medium. Growth is usually plotted as the logarithm of viable cell number versus incubation time, and the growth curve usually has 4 distinct phases. Here we have a microbial growth curve in a closed system. The 4 phases of the growth curve are identified on the curve and discussed in the next few slides. So let's look at the 4 phases here. First we have the lag phase, followed by the exponential or log phase, the stationary phase, and the death phase. Note the axes, we're looking at the log number of viable cells over a period of time. The first phase of the microbial growth curve is the lag phase. Now let's consider when microorganisms are introduced into new medium, usually there's no immediate increase in cell number, and hence why it's called the lag phase. This is because the cells are normally synthesizing new components. For example, the cells may be old and depleted of ATP, essential cofactors, and ribosomes, and these have to be synthesized before growth can begin. The medium that you're using may be different, even slightly different from the previous one, so new enzymes have to be synthesized in order to use different nutrients. Whatever the causes, the microbial cells must retool, replicate their DNA, increase in mass, and finally divide. The second part of the growth curve is the exponential or log phase. This is when the microbial cells are growing and dividing at the maximum rate possible, given their genetic potential, the nature of the medium, and the conditions under which they're growing. Their rate of growth is constant, that is, they're dividing and doubling in number at regular intervals. But because each individual divides at a slightly different moment, the growth curve rises smoothly rather than in discrete jumps. The population is most uniform in terms of chemical and physical properties during this phase. So exponential phase cultures are normally used in biochemical and physiological studies. The third part of the growth curve is the stationary phase. This is where the growth curve becomes horizontal because the total number of viable cells remains constant. This may occur because metabolically active cells stop reproducing, or it may occur because the reproductive rate is balanced by the death rate. So what are the possible reasons for entry into the stationary phase? Well, the first possible reason here, nutrient limitation, is a fairly obvious one. But what about oxygen availability, particularly for aerobic microorganisms? Oxygen's not very soluble, and it may be depleted so quickly that only the surface of the culture will have enough oxygen for growth. Toxic waste accumulation. For example, some bacteria produce so much lactic acid and other organic acids from fermentation of sugar that the medium becomes very acidic and this inhibits growth. There's also some evidence that growth stops when some critical population density is reached. So bacteria in batch culture may enter the stationary phase due to starvation, and this probably occurs widely in nature. But microorganisms have evolved a number of strategies to survive starvation. So we know already that some species of bacteria will form endospores when environmental conditions are not positive. Other species of bacteria do not form endospores, but they undergo other morphological changes. They may decrease in cell size, the protoplast may shrink, and they may undergo nucleoid condensation. Other microorganisms have been shown to produce what are called starvation proteins. These proteins make the cell much more resistant to damage in a variety of different ways. These are proteins which will increase cross-linking of peptidoglycan and cell wall strength, proteins that protect the DNA, prevent protein denaturation, and proteins that assist to renature damaged proteins and damaged DNA. So as a result of these mechanisms and other mechanisms, the bacterial cells are harder to kill, and some important pathogens become even more virulent when starved. So clearly this is of great practical importance in both medical and industrial microbiology. The fourth part of the growth curve is called the death phase, and for many years the decline in viable cell number was simply described as the death phase, and it was assumed that cells simply started to die, as even when they were transferred to fresh medium, no cell growth occurred. The problem is that the loss of viability was often not accompanied by loss in total cell number. So it was then assumed that the cells were dying but not lysing, which is what would normally occur. This view of cell death is currently under debate. There are two alternative hypotheses, which you can see here. The first is proposes that the cells are viable but not culturable, VBNC. The second hypothesis proposes that programmed cell death, or PCD, is occurring. Let's look at these two on the next slide. These hypotheses are illustrated on this slide. So in A, we can see the traditional view, which assumed that as cells leave the stationary phase due to starvation or toxic waste accumulation, then the decrease in population number is simply due to cell death. In B, some people believe that a fraction of the microbe population dies due to activation of programmed cell death, or PCD genes. Then the nutrients that are released by dying cells supports the growth of other cells. In C, the viable but not culturable, or VBNC hypothesis, proposes that when cells are starved, they become temporarily non-culturable under lab conditions. Then when they're exposed to appropriate conditions, some cells regain the capacity to reproduce. The generation or doubling time is simply the time required for the population to double in size. And this varies depending on the species of microorganism and of course on the environmental conditions. The range is from 10 minutes from some bacteria to several days for some eukaryotic microorganisms.